Health Canada's ongoing COVID-19 vaccine safety claims deserve more intense examination following alarming DNA contamination findings in a first-of-its-kind, peer-reviewed Canadian-led study. If you don't know what I'm talking about, let's dig in. Health Canada has been pressed in official government communication on the safety and quality controls surrounding lipid nanoparticles called LNPs used in mRNA-based vaccines for COVID-19 like Pfizer and Moderna's, their related boosters, as well as the lipid-based drug Onpattro. The parliamentary exchange was prompted by Conservative MP Dean Allison, whose detailed questions probed potential mutagenic risks from vial impurities. These include things like residual solvents, metals, and elemental contaminants. Allison also asked for assessments of the LNPs themselves, including the use of polyethylene glycol called PEG, that component, and risks of complement activation-related posito allergy, called CARPA. Now, CARPA is a non-allergic hypersensitivity reaction to the complement system, a part of the immune system that enhances antibody responses and clears pathogens. It can be activated by certain substances, such as drugs or nanoparticles, like those used in mRNA-based vaccines, i.e. the lipid nanoparticles, or PEG, the polyethylene glycol. A reaction of this kind can lead to symptoms like skin rash, flushing of the skin, shortness of breath, or anaphylaxis-like responses. Unsurprisingly, current government guidance on contradictions and precautions of these vaccines fail to mention this reaction risk at all, nullifying informed consent. That didn't stop Parliamentary Secretary Maggie Chee, responding on behalf of the Liberal government, from emphasizing Health Canada's stringent regulatory oversight. She assured that impurities in LNPs were evaluated per International Council for Harmonization guidelines with levels of solvents and elemental impurities falling below permitted daily exposures and classified them as non-mutagenic. The response stated that LNPs were characterized and controlled through various tests, with PEG deemed non-mutagenic as well. But these claims were made based on non-clinical studies conducted on novel lipid excipients. Shockingly, no complement-related safety testing were required or requested, since there's no established test for PEG hypersensitivity. For Onpattro, similar assurances were given, with impurities said to be below the acceptable limits. This official narrative paints a picture of thorough, guidance-compliant reviews ensuring safety, but just days prior to this response, a peer-reviewed study published in Autoimmunity has thrown claims of stringent regulatory oversight into question by revealing staggering levels of residual plasmid DNA in the very vaccines Health Canada deems acceptable. Led by molecular virologist Dr. David Speaker alongside Kevin McKernan and Dr. Jessica Rose, the research quantified DNA contamination in vials from Ontario, Canada, using fluorometry and qPCR. Findings showed total DNA exceeding the FDA and WHO's 10 nanogram per dose limit by an astonishing 36 to 153-fold for Pfizer and 112 to 627-fold for Moderna. Critically, Pfizer vials contained the SV40 promoter enhancer sequences, with contamination levels ranging anywhere from 0.25 to 23 nanograms per dose. This viral element linked to potential genomic integration was absent in Moderna's vials, likely due to differences in manufacturing processes. The study shows just how sweeping methodological flaws in the regulatory testing are, noting that DNA is encapsulated within LNPs, shielding it from degradation and enhancing transfection efficiency. This encapsulation, the authors warn, amplifies risks. Moderna's patent states that a genetically engineered DNA packaged in a lipid nanoparticle is directly injected into a living host and could cause insertional mutagenesis and activation of oncogenes or the inhibition of tumor suppressor genes. Preliminary VAERS data from the United States analyzed revealed correlations between these high DNA levels and serious adverse events, with Pfizer lots, some of them reporting up to 941 serious adverse events. These revelations directly challenge Health Canada's assertions. While the government focused on lipid-specific impurities like solvents and metals, it seemingly overlooked or downplayed broader DNA contaminants, which the study ties to unquantified health threats, including cancer and prothrombotic effects from repeated dosing. The lack of assessments for PEG hypersensitivity further highlights gaps in evaluating LMP-related risks and the vast unknowns about nanomedicine. As post-vaccination health concerns mount, this is yet another discrepancy that demands answers. Why weren't DNA levels scrutinized more rigorously? Independent researchers like Speaker McKernan and Rose's work expose what official reviews have missed or maybe minimized. It's time for transparency. Halt boosters until these risks are fully addressed. For Rebel News, I'm Tamara Ugolini. The more that is learned about these shots, the more it seems like they should probably be pulled from the market. If you agree, sign our petition at nomoreshots.ca. There you can also sign up so that you never miss a report on this topic. And of course, support this kind of investigative journalism if you so please. That's nomoreshots.ca.